I'll be talking about CASARETT's classification of radiosensitivity. So this divides the cells from most sensitive to least radiosensitive group of cells. It is divided into four classes: vegetative intermitotic cells, differentiating intermitotic cells, reverting post-mitotic, and fixed post-mitotic. So vegetative intermitotic cells are the most radiosensitive, and fixed post-mitotic cells are the least radiosensitive. So vegetative intermitotic cells are the cells, like for example, it are the cells which are present in the basement, in the basal epithelial layers, like skin and intestinal crypts actually, and some of the undifferentiated stem cells and germ cells. So the cells which are present in the basement membrane, they're constantly dividing. So they're constantly dividing, but they do not have differentiation. So usually from the basement membrane, as the cell matures and moves up in skin, then they become a differentiated cell. So in the cells in the basement epithelium, they are usually constantly dividing, and there is no differentiation in these cells. So those are vegetative intermitotic cells, and this group 1 are the most radiosensitive cells. Examples for those are skin, intestinal crypt cells, and undifferentiated hematopoietic stem cells and germ cells. Next is differentiating intermitotic cells. So for examples for these are myelocytes and spermatogonia. So so as the name suggests, it is like differentiating. So some of the cells which constantly divide, so form they divide for a few cycles, they divide for a few amount of cycles, then they become a differentiating cell. Then it is not dividing. So for first few cycles it will divide, then it will become a differentiating cells which will be non-dividing. So so this includes all of the cells that are intermediate between the stem cells and the differentiated cells. So stem cells and the various process which to become a differentiated cell. So any cells which come in between all of these are differentiated intermitotic cells. Example for those are myelocytes and spermatogonia. Third type is reverting post-mitotic cells. So these are the cells which usually do not divide. So division does not happen in these cells. Like normally they don't divide, but they have potential to divide. For example, cells in the liver, and cells in the pancreas, cells in the kidneys, and sometimes adrenals and thyroid. So here normally the cell does not divide, but if it requires, it has the potential to divide and revert back to intermitotic cells. It has the potential to divide. So all of these cells are reverting post-mitotic cells. Next is fixed post-mitotic cells. So these are like, they do not divide at all. They are permanently not dividing, permanently non-dividing cell. So example for these are nerve cells and our skeletal muscles. So so these are like permanently non-dividing cells. There are some short-lived cells also which come into this into this class. Those are neutrophils and RBCs and some superficial epithelial cells. So these are the four main classification of CASARETT's classification. So there are some exceptions for these. The first exception is that connective tissue stroma, like fibroblast and endothelium, these are like intermediate between the second class and the third class. Second class is DIM, that is differentiated intermitotic, and reverting post-mitotic cell. Now another exception is that peripheral blood lymphocytes. So lymphocytes, they come into the fourth class. They are fixed post-mitotic cells, but they are very radiosensitive. So this is like one of the exception for this CASARETT classification. Even though the lymphocytes, these lymphocytes now further do not divide, but they are short lived. They come into the class of fixed post-mitotic, but they are very radiosensitive. That's one of the exception of FPM. So other peripheral lines like granulocytes, RBCs, and platelets, those are very radio resistant. That comes here. These are platelets also. These are very radio resistant compared to all of these cells. So the latency time of cytopenias after radiotherapy is based on the lifespan of these differentiated cell. That's it.